Do we have any insights into other sort of longevity interventions that people think about and their impact on oral health in either in preclinical studies in mice or in people? And I mean, it's kind of like the list that everybody's going to think of: metformin,
Yep.
or NAD precursors,
Right.
or alpha-ketoglutarate, or caloric restriction, other dietary things. Do we know of anything that is effective or where there's even a hint? Or maybe that goes the other direction?
Yeah. Not that I'm aware of, but I could tell you and you always say this, what's a really low hanging fruit is you go back to all those ITP studies, kind of what we did in our first
So
study.
just ITP, mouse interventions testing program.
Yep. Mouse intervention testing program. And kind of what we did with the rapamycin was just go back to all those interventions.
Yeah.
They have everything stored.
Yeah.
And see if any of those interventions actually have an impact, like metformin, right? Patients are diabetic, right? And they are taking metformin, so they probably have some sort of gum inflammation already underlying there.
Right.
So maybe they can look at those mice that have taken metformin or other, compounds that they have. But I think you'd have to be cautious because, a lot of those compounds, at least from the ITP, only certain compounds do extend lifespan, right?
That's right.
So it's not every single compound.
But it is possible that some compounds might not extend lifespan. I mean, metformin's a good example.
Right.
In the ITP, metformin did not extend lifespan, but there's a whole body of evidence that metformin does have impacts on metabolic health and potentially some other age-related diseases or functional measures.
Right.
So it might be the case that it would have an impact there. It's interesting. I mean, it begs the question, like, why hasn't that been done?
Maybe they just don't have a dentist on their team.
Yeah, or it's because they couldn't get a grant to do it. I mean, the things that get funded or don't get funded by NIH, don't get me started. But no, it seems like really smart. And we'll talk a little bit about that mouse work that you were referring to with rapamycin, where we looked at teeth from the mice treated with rapamycin. I think a good starting place might be to just go back briefly to that conversation we had in 2015 when you came to my office and you were sort of convincing me why I should pay attention to the mouth, and just refresh for our viewers, what is the connection between aging and oral health?
Yeah. So with age, a lot of the tissues in the mouth decline, like the functionality of it. Cell processes decline, and they happen very slowly. And so, and because of that, you have increased risk for like periodontal disease or gum disease, or, like you said, salivary dysfunction, cancer, and that leads to subtle changes like taste bud changes. And so, and we follow this all the time, and patients know about this, right? But the downside right now is, as dentists, we have no idea how to actually treat them or actually how to regenerate some of these areas to go back to where they were. Because once you have periodontal disease, you have periodontal disease.
Yeah.
And so there is that strong relationship, both biologically, but also clinically we see this, but there's really not much that we could actually do right now. Yeah.
And, back in 2015, when we started down this path, what was sort of the state of the understanding of the research at that point? Because I get the impression that, like you said, of course, dentists kind of knew that old people have more oral problems, just like medical doctors know that older people have more medical problems. But what was kind of the state of scientific knowledge at that point?
Yeah, I mean, if I can cautiously say this, I think people, and this goes back to part one where we talked about really good science, where people talked about aging, but it wasn't really actually aging. And we actually published a small little review on this where people would say, well, these aged mice have this. But when you actually look at the aged mice ages, right, they're comparing like a 3 to 4 week old mice to a 12 to 14 week old, right? So
Classic
they are
mistake.
Yeah, so they're all old. So I think this goes.
So maybe this is worth just taking a minute to really talk through like what is normal aging in a mouse, like in terms of actual chronological numbers, right? So what would you consider sort of young mouse, middle-aged mouse, and old mouse?
So a young mouse, probably like 4 to 6 months, right?
And mice, again, develop pretty rapidly, right?
Exactly,
so they're sexually mature by what age? 4 months.
Yeah,
certainly.
probably even before that.
Okay.
Yeah. And then, middle age, you're looking at probably like 13 to 16 months, like in that range, like 12 to 14. I mean, the range is always a little bit different.
So again, middle age is a tricky term too, right? But so that would be maybe the equivalent of what, a 40-year-old person?
Yeah, 35, 45, kind of in that range. And then I've heard certain terms, like there's pre-geriatric and there's geriatric mice, right? So if you want to go that way, like maybe like 18, 19, 20, that months range is like your basic pre-geriatric. And then after 21, 22 months, then you're kind of going on to geriatric. And then some groups will say it's actually after 24, 25.
Yeah, I would
months,
actually push back on that and say, you know, if you're thinking like the geriatric equivalent in a human, it's got to be at least 24, maybe closer to 27, 28.
Right. So when you start the treatment, like at least 24, 25.
Yeah. But I always think of like a good rule of thumb is 20 months in mice is about 60 years in people, ballpark, biologically.
That's kind of how we kind of still define it in our lab too. Yeah,
okay. And if anyone's confused, there's an episode on chronological age versus biological age. So I'd encourage you to go take a look at that episode and I go into a deep dive on what that means. But okay. So I think what you were saying though is that, you know, at the time we started really looking at this area, there were only a few studies and most of them were they were calling them old mice, but they were using mice that were really maybe 35 equivalent of 35 years old in people.
And the terminology of age and geriatric, especially when in animal studies, was not being utilized correctly from kind of the aging field sense, right? And I think that's where in the lab, the synergy worked out because, you know, you knew everything about,
would not,
you know, most of the stuff.
Nobody would agree with that statement.
Right. Well, like at least from my end, right? Like you understood kind of the aging biology part. And so, and maybe we'll go into this too, but you know, all the ideas I had to, for example, with periodontal disease, right? Like I remember in the first few meetings, you know, you asked the questions like why are we inducing it? Have you just looked at old animals, right? And so that really got me thinking. I was like, wow, we actually have been looking at this scientifically, you know, and many of the data, sure, they're good data, but it's not relevant to kind of the actual aging biology that's happening.
Right. And this is just very, very common in other fields as well. Again, I think cancer biology is sort of a classic example here where people try to study an age-related disease in a young animal and you miss all of that altered physiology that goes along with aging. And I honestly think that's why, you know, people always blame mice as the reason why a lot of drugs that work in mice don't work in people. I actually think it's not the mice. It's the fact that most of those studies were done in young mice when this is a disease of old people.
Exactly. Yeah.
And so that's one of the reasons why I've always felt more comfortable with the idea that things we learn about biology of aging in old mice are actually pretty likely to translate through to people.
Absolutely. Yeah. I mean, you know, maybe I'm jumping ahead a little bit, but like even periodontal disease, my idea of coming in was inducing that disease in young animals.
Yeah, I remember.
Because that's what we've been doing, right? And this is where it goes back to my point of having a good, you know, a science lab that really does rigorous science. Because your first point, and maybe it was not verbatim, was like, why aren't we just looking at old animals, right? And I understood that now, and even then was that, yeah, the disease is there, but the complete underlying biology is it's just missing.
Right.
So, and that's also why a lot of periodontal disease, you know, compounds that have been developed or drugs that develop, you know, maybe has not worked because you're only targeting a few things, or maybe you're just targeting whatever happens in a younger animal.
Right.
So at the time, back to your question of, you know, we haven't really delved in. Aging was there, but there was not good science to back it.
So at that time, had anybody actually really characterized the change in the oral cavity with age in mice?
There were a few studies that did that, but it was just more of, okay, these old mice also develop, you know, periodontal disease, and that's it.
Yeah.
It was very basic studies. No molecular, no nothing. It was just kind of an observational, because at the time, most people were thinking, oh, aged animals, I actually have to age it out for 2 years.
Right.
And
that's actually, to be fair, I mean, I was sort of, you know, a little bit maybe too dismissive of the cancer research done in young animals. To be fair, the reason why people do that is because you could do it a lot faster. You don't have to age the mice out to 24 months. Now you can commercially buy 24, 28-month-old mice. There's really no excuse, yet people are still doing those kinds of experiments in other fields, but that's maybe a different topic. So what about non-human primates? I assume they get periodontal disease with age. Had anybody really studied it there? Or is it the same problem where, you know, you don't really use aged non-human primates in research?
Same thing where they will use like we talked about in part 1, where they'll use like a ligature model or a silk model to induce that
in young
disease
animals.
in young animals.
Yeah.
And I know I think there was 1 or 2 studies that actually did it in middle age, and they profiled kind of the inflammatory changes. But the data is out there. But really the question was, okay, like they're just publishing kind of these changes. There's no more, there isn't more science kind of beyond that, right? It was just like, oh, these are the inflammatory changes. Just characterize it and then just kind of leave it there, right?
Yeah. And then I assume nobody's ever really, I guess I would probably know if they had, because I work on the dog aging project, but I'm thinking about companion animals. Like we all know, if you've ever had an old dog or an old cat, that oral health declines with age. And I would venture to say that most companion animals don't get regular dental checkups. Some do, but most don't. So we've known that it's sort of been it is an age-related phenomenon. But again, I don't know if anybody's ever really carefully looked at that, even from a characterizing perspective. Like, what's the frequency of these things? And
speaking of companion dogs, like we know, like cats and dogs, like number 1 thing they get is periodontal disease, right? And then going back to your point, non-human primates, like we actually see, like now our group, we're seeing that as the animals do get older, they do develop natural periodontal disease, just like mice. And even not only facility raised animals, but if you go out in the wild too, wild non-human primates also develop.
Yeah, I was going to ask about wild or zoo animals too. It'd be sort of another place you could imagine looking. So I assume, one of the things about the wild is not every species in the wild lives long enough to make it to old age. So mice are a good example. In the lab, a mouse will live to be 3-4 years sometimes. In the wild, they rarely make it through a single year. And so they don't have the opportunity to uncover these age-related diseases, and periodontal disease falls into that category. So I guess let's think about now, at the time you started your thesis, sounds like there have been a couple of papers that had kind of characterized the fact that mice in the laboratory develop periodontal disease. What did you set out to do to kind of build on that? How did you start to try to approach this problem?
So I kind of knew in that background. So I actually approached it from kind of the aging biology perspective of, okay, what do we know in the field that actually works? Like what is the pathway? Like as any graduate student, you're in literature all the time. And so what are people actually looking at here? And what is that kind of the area that, people are interested in? And for us, and for me at the time, it's like, is there a compound out there? And this is before I knew work with rapamycin or, what is the compound that actually, there's got to be people that show that this works, right? Some compound extends lifespan, health span, et cetera. And so while doing the literature search, I found rapamycin. And what was interesting about rapamycin is I knew that as a dentist, like rapamycin was immunosuppressant, kidney transplant get this, they get oral mucositis. We could talk all about that later. And what was interesting was if you actually look in the literature, like I found like a couple hints where one was, rapamycin actually alters bone turnover. So there were studies that showed that it inhibits osteoclast levels in osteoclast or bone resorbing cells. And then there's other studies like even your lab that showed that, it alters the gut microbiome. So the microbiome itself, right?
Because that was right about the time that you were joining the lab, right?
Exactly,
that paper came out. Yeah.
And so as any other graduate, I had all these projects that I was working on. And so I knew it altered the microbiome. And then there's other reports that showed, changes in inflammation. And so like those targets, right? Inflammation, microbiome changes, bone turnover. I was like, this is what periodontal disease is. And if periodontal disease is age-related, has anybody looked at rapamycin in these old animals that live longer? And if they actually rapamycin, if it extends their lifespan, do they actually have less periodontal disease? It just kind of fell together like that.
Right. Yeah. And I remember when we were talking about this, and I think, one of the ways I thought about it was, there's a hypothesis, because it hadn't been formally proven, and maybe you could argue it still hasn't, but there was a hypothesis that periodontal disease was driven by the biology of aging the same way that other age-related diseases are, right? And I say it's a hypothesis. It almost certainly correct. But one of the ways you test that is to modulate the biology of aging, slow the biology of aging, and the prediction is that if the model is correct, then you should slow periodontal disease, right? So that's the way I thought about it. And then, of course, the obvious thing to test was rapamycin, because it was kind of the gold standard at that point. So So you designed a project to like try to start testing that. Your first paper was titled Rapamycin Treatment Attenuates Age-Associated Periodontitis in Mice. So I think we know the answer, but we'll still go through it. But let's kind of go through this study and talk about like what was actually done. So this figure here is, I think it's part of figure 1 from that paper. So maybe you can just kind of walk us through what is being shown here and how did you tackle this problem? Because I thought it was actually a pretty neat way to approach this.
Yeah. So this was actually from this technique. What you're looking at is called a micro CT. And basically, you know, it's just like any other CT scanner, like in the medical field, but it's a micro. So you can do these in small animals. And this
is X-ray based for people who might not know what a CT scanner does.
So basically, you take, you know, small slices, sections of, you know, each area, and you have thousands of hundreds of photos that get combined into this 3-dimensional image, a 3D image. And this technique I actually picked up during one of my rotations in that craniofacial genetics lab I talked about, where one of my first projects was looking at the retinoid pathway and how it relates to cleft lip and palate. And so I was doing these scans and learning how to do that on there. But what was also interesting was at the time, I wanted to look at periodontal disease. And in the clinic, the way we diagnose periodontal disease is actually probing around your gum pocket. So when you go to your office, you know, you hear 2s, 3s, 4s.
Pocket depth.
Yeah, pocket depth. We're reading that. So I was thinking, okay, well, how can I actually look at periodontal disease in mice? Because I can't take a little probe in there. And I thought, okay, I could just image them. And if the image is 3-dimensional space and we know exactly the measurements, we could go in there on the computer and just measure kind of the bone loss.
Right.
So what you're looking at here is a figure from a young animal on the left, and this is the top teeth of the mice. And on the right, you're looking at an old animal where you could clearly see kind of, we talked about grinding earlier, but mice do grind. So you can see the grinding patterns and the clear bone loss that just happens between the areas that are marked in the yellow.
Yeah, so the yellow lines are a distance measure from 2 landmarks, right? Let me see if I can remember this. CEJ is cemento-enamel junction, and ABC is alveolar bone crest, right? So as that distance gets larger, that's telling you that the bone has sort of receded downward or upward, depending on whether you're at the top or the bottom of the mouth.
Exactly, yeah.
Okay, so in this case, the numbers, when they're larger, mean bone loss. And these are C57 black 6J mice, which is just important. I mean, I don't want to get into the weeds of the different mouse strains, but when you're talking about mice in the laboratory, it's important to appreciate that there are different flavors. C57 black 6J is one of the most commonly used inbred lab strains. And one important thing to appreciate about C57 black 6J is because it's an inbred lab strain, it's what's called genetically homogeneous. All of the individuals are essentially genetically identical. I'm going through all this because we talked about the ITP earlier, and we're going to talk about UM-Het3, which is a different mouse strain in a minute. So C57 black 6J, young are 2 to 3 months old, old are about 24 months old. So we're, again, ballpark 70-year-old, 65-year-old person biologically. Okay, so there's clear loss of bone around the teeth. I think that was sort of known, but maybe never quantified like this carefully beforehand.
Yeah, everything was quantified manually using a microscope at the time.
Got it. And so then in the next figure, this is showing the effects of rapamycin treatment, again in C57 black 6J. Take us through what the treatment was here.
Yeah. So I may have to step back a little bit. So I think this is where, because I had all these lofty ideas as a new grad student, right? We're going to buy this, we're going to buy that. We're going to.
And I was like, wait a minute, we don't have a grant to do this yet.
Exactly. So I think this.
By the way, we never got a grant to do this. Thank you very much, NIA.
Among many other things, right? So I think just a backstory about this. So when we thought about this, we're like, okay, is there existing studies that are being done that have the head pretty much, or that we could look at the oral cavity and evaluate kind of our hypothesis.
Right. So the idea here is we don't have the money to do the experiments ourselves. Maybe we can find someone else who already did something with old mice, and we can just get the teeth or the jaws and measure them. Yes.
Right. Yeah. So this study is from I think it was from Ellen Quarles' study where they published on
in Peter Rabinovich's
lab, where they published the report on cardiac function in old mice. And so these are from Ellen's cohorts, and so these older animals that are treated with rapamycin were on 14 parts per million rapamycin.
Right.
So that's the dose that Ellen Quarles gave to the old animals.
And just for people who may be kind of in the weeds, this is a encapsulated form of rapamycin called E-Rapa added to the food. 14 parts per million is the amount in the food. And what that translates to, I don't know off the top of my head, but this was the amount or the dose that was used in the first interventions testing program study in mice. So it's kind of like the baseline dose I think most people in the field will refer to in mouse studies. Okay, so 14 parts per million in the food. The mice had been treated for 8 weeks.
8 weeks,
And this was initially a study to look at heart function, where they did see improvement in heart function by rapamycin. And then they had kept the bodies of the animals after sacrifice.
Yep.
Okay.
Yeah. And so, I mean, I still remember the first thing we asked was, do you still have the heads?
Right.
And so like, oh yes, we have the heads. So we went in there and collected. So this data, what you're looking at is we basically took those measurements we talked about earlier. And at the time, as a graduate student, one thing that all of us had in the lab was actually we actually support a lot of undergraduate research. And so I had 2 undergrads that worked with me. So I basically blinded all the samples and I said, okay, these are the metrics that you're going to use to measure this. Please measure it. And then this is the combined data where you're seeing that versus the old animals where you see high numbers, which means there's a large difference.
More bone loss
compared to the rapamycin treated animals, which has less bone loss overall.
Right. Yeah. And just because, when we talk about the next paper, to be clear, these are not, so when you go from old to old with rapamycin, these are not the same animals before and after. These are different groups of animals who either got kind of like the placebo version of a placebo in a mouse, we call it a vehicle, or they got rapamycin, right? Okay. Yep. And the mice that got rapamycin have less bone loss. But I mean, I think looking at this, there is a statistically significant difference. They had less bone loss. It's not huge, right? But it is significant. And they only got the rapamycin for 8 weeks. So, you might expect that if you had it for longer, you would see more. But this was kind of
a higher dose, right?
Yeah, or higher dose, right? But this was kind of a first, I think, indication there's something interesting here potentially.
Right.
Okay. So then what happened next?
So then what's the next one? So then we talked about ITP earlier.
Yeah.
So then it goes along. We didn't have money to do this. We didn't have the grant. Okay. I'm pretty sure people store things, especially the ITP will kind of store these animals. So Matt reached out to the ITP and said,
specifically to Rich Miller, who is one of the PIs. He's the PI at the Michigan site for the interventions testing program.
Yeah. So we asked Rich Miller, hey, we have this really kind of interesting result. And is there a way we can get the animals from that ITP study that you have still stored? And can we look at those animals?
Yeah. And I just want to give a plug for the ITP because I think there are many great things about the ITP. I'm a huge proponent of continuing that program. I think it's been hugely important. But one of the things they do really well is they store tissues, they biobank tissues, and they are very, very willing to give those away to other investigators to support other types of studies. So we were very fortunate, to be able to get access to these animals. And this is the point I think that we were talking about a little bit earlier, where the same thing that you did here with the rapamycin treated mice could be done with ITP mice for every other drug that extended lifespan.
Right.
Really interesting question. Somebody should do that someday. So anyways, we got mice from Rich. So go ahead, take us through this.
One thing too, I wanted to also thank Rich was that, when we presented it, he was obviously excited about it, but also because of the lifespan, right? And this is kind of, we had to pick the right animals that kind of died around the same age to control for that.
Right.
And I remember Rich bringing that up and saying, okay, to do this, we have to actually, and you mirrored this too later on, early on too. And so he kind of went out of his way to actually find those animals.
Let me make a point here because I think it may not be clear to people who are watching this, is that in the study we just showed from Peter Rabinovich's and Ellen Quarles' mice, those mice were sacrificed at probably 24 months of age or something like that at the end of the experiment. So they were sacrificed. The mice from the ITP died naturally, and then they store the biosample. So it creates a different complication in the experiment when you have mice. Some of the mice died at 24 months, some at 26 months, some at 28 months, some at, in this case, 35 to 37 months. And one of the questions is, are they dying for different reasons at those ages? We don't know. But yes, so Rich made the point that we want to be careful to make sure that we're selecting mice that died at about the same age range.
Right. And so and that was, he could have just said, yeah, here's the animals without really giving us that, extra thing. So I wanted to think. So anyway, so what you're seeing here is very similar to what we showed you before. Is we took these old animals, and it was very clear from these UMHET3s, this mice that the rapamycin was actually doing something. Because, and this is just one image that I'm showing you, but what we're finding is that the old animals, and obviously we didn't have the young UMHET3 animals in this case, but the old animals had that bone loss, which is again the higher number there, and all of the animals that were treated with rapamycin lifelong, starting at I think 9 months, showed that still that significant attenuation, or less bone loss overall. And so and that's kind of what you're seeing both in the imaging as well as the data.
And a couple of points to make again. So we talked previously about C57 Black 6J. The interventions testing program uses UMHET3 mice. The reason why that's important is the UMHET3 mice are, instead of being inbred, genetically heterogeneous. So they're a mix of 4 different genetic backgrounds. Every individual is genetically different from every other individual. So many people, including Rich, who's very vocal about it, feel that the UMHET3 are a superior mouse model for studies of aging and a variety of other things because of that genetic heterogeneity. I think what's really nice here is we're seeing the same impact of rapamycin in 2 very different genetically mouse backgrounds, 2 different doses. So this is 42 ppm, so a higher dose. Also, this is different in the sense the mice were treated lifelong, starting from 9 months of age, whereas in Peter and Ellen's study, they were only treated for 8 weeks, starting in middle age. Again, what we don't really have is a way to compare the same mice before and after rapamycin treatment. But we'll get to that because I think that's obviously one of the important questions that remained after this study. So in any case, I think when I saw this, I was pretty convinced, and I know you were, that okay, this is real, right? Rapamycin is at least attenuating bone loss, slowing it down in mice, and I think we can make maybe not in all genetic backgrounds, but at least in 2 very different genetic backgrounds. So very reassuring to see that. It seemed to be shared across the different genetic backgrounds. So I think then at this point, there's a couple of questions you could ask, right? Is it only affecting bone loss, or is it affecting other things in the oral cavity? And I think a particularly interesting question is rapamycin just slowing the loss, or is it doing something else, right? And so then I know you set out to try to answer those questions in the next study. And I think you convinced me that even though I didn't have a grant to fund this, and never did get a grant to fund this, thank you very much, NIA, despite trying over and over and over again, that we could somehow figure out a way to scrape up enough resources and find cool collaborators. And so I think this allowed us to do what turned out to be a really cool study. So the second paper of yours that I wanted to talk about, really, I think tried to answer both of these questions. And the title of that paper was Rapamycin Rejuvenates Oral Health in Aging Mice. So you keep giving away the punchline in the title of the paper, but still, I think it's worth going through this to set up for people like what was done and what we can conclude and maybe what we can't conclude. So this really, I think, just kind of sets up the design. So maybe take us through what was done here. And I mentioned finding some cool collaborators that allowed us to do this. So maybe talk through what I was referring to there as well.
So it goes back to the same repertoire, right? We didn't have funding, didn't ever get funded. And so we were trying to figure out, okay, how do we plan the next cycle? And so around the same time, I was able to, we were able to get some older mice because I was able to apply for a fellowship through NIDCR.
And just for people who may not be in the weeds on NIH, so NIA, which I keep mentioning in passing, is the National Institute on Aging. That's where the vast majority of biology of aging research is funded through. That's one of the branches of NIH. NIDCR is the National Institute of Dental and Craniofacial Research, right?
Yep.
So that's kind of the dentistry division of NIH, right?
Yeah.
Okay.
And so I applied for dual degrees. It's called an F30 fellowship. So I applied for that fellowship.
And that just meant I didn't have to pay your salary anymore. And you got a small amount of funds for research.
So I helped out in some way. But getting that fellowship allowed me to do was get animals from the NIA, because I specifically put in aged animals in that grant.
Right. So at this time, and I think it still exists,
Yeah.
National Institute on Aging had an aged rodent colony, but not everybody could get access to it. Only people who had NIH-funded grants to study aging biology could get access to the aged animals. And I believe back then, we didn't have the money anyways, but I don't think old mice were... This was probably right about the time they started to become commercially available. So I don't think they were widely commercially available yet.
Yeah.
Okay.
So we were able to get those animals through that. And then right around the same time, and I don't know, I still don't know exactly how that happened. It just kind of merged together where we got this opportunity with the Jackson Labs.
Right. So that was a case where we mentioned briefly the Nathan Schock Center in the last episode. So Jackson Labs has a Nathan Schock Center, and Nathan Schock Centers are mandated to give out pilot projects. And so the Jackson Labs Aging Center, which is sort of an umbrella over their Nathan Schock Center, has done this really smart thing where they have used Nathan Schock Center resources and Aging Center resources to make aged animals available for experiments to the whole community. And people can just, there's a competitive proposal process, but people can propose an experiment, and if it seems like it's going to have an impact, then they'll go ahead and do the experiment there, often on the animals. So I knew the PIs of the Nathan Schock Center and proposed this experiment, or maybe you even wrote the proposal.
Yeah, so we wrote the proposal up, and it just happened. I still don't remember exactly at what sequence it came, but it happened kind of simultaneously where we got the NIA mice, and we got this Jackson pilot. Yeah. Let's just do both.
Yeah. Right? And I mean, this is often how it goes when you don't have grant funding. You leverage resources. And I mean, even still getting the mice, it's still a pretty expensive experiment, but that allowed us to be able to do the experiment when we wouldn't have been able to do this if we weren't able to leverage these resources.
Yeah. And the neat thing with the Jackson, with the Jackson collaboration that we had was, I mean, there were other researchers that were interested in kind of understanding how rapamycin was working. And the additional thing we were able to add on to those cohorts, and we show it here, is that we actually took a dental CT or micro CT before we even started the rapamycin. And then...
And you would anesthetize the mice, put them in the CT scanner, and then they'd come out of the anesthesia and go on about their lives.
Yep. And then they'll get rapamycin, and then right before we had...
vehicle,
right? Yep. The placebo of that. And then right before the tissues were collected, we took a final micro CT just to, and the idea here is to see, okay, we know they're going to have bone loss. Does that bone loss change in that same animal? So in the previous study, if it was looking at what this animal would go through the study, another cohort would go through a study, and we just compare. This is actually looking at the same animal over a course of time. Right.
Much more powerful.
Right. And then with our the NIA animals, which we raised here in Seattle, we just took the micro CT kind of afterwards, right? We couldn't do any of the what we call longitudinal kind of CT scans. So we had a cohort of animals in Seattle, another cohort of animals at Bar Harbor, Maine, doing the same exact treatment, and after 8 weeks, us going down there and collecting it.
Right. Yeah. And this was with the 42 parts per million. But this time in C57 Black 6J, right? So it's a higher dose than what was in the first study with that strain background, right? But the same dose that was in the Het3s from the ITP.
And I don't know if you remember this, but when we were designing this, we're like, okay, because there were 3 dosages that ITP tested. I think it was like, where was it? Like, I know 14 was 1.
It was 1/3 of 14, so 4 point whatever.
Right.
6.
And so, and I think this was your suggestion was, well, the highest dose seemed like it's extending the lifespan.
The most.
So let's just go with the highest dose. So we just went with the highest dose.
And we now know that this isn't even the top end for lifespan extension, but it's the highest dose, I think, still that the ITP has tested, and where they see the biggest lifespan extension at that dose. So that was the rationale.
And then we sacked them around 22, 23 months. And this time, compared to the previous study, we were able to collect like gum tissue, bone tissue.
So not only do you get the bone measurements, but you can get tissue measurements,
even microbiome, which we'll talk a little bit about later. Yeah.
Okay. So that's great. Okay. So what did you find when you looked at the bone loss first, right? Because that's what we knew from the prior studies. We should see a difference between the rapamycin treated mice and the vehicle treated mice, but we'd never before been able to look at the same animal before and after.
So, so on here, what we found was in the same animal, the areas that showed bone loss, there was actually gain of kind of hard tissue or bone in that area.
Isn't that amazing? I mean, even still, like I know this data. And it's like, to me, that is just like the most amazing result.
And it's not, I mean, so if you really look at the picture, what was really neat to me was it wasn't like everywhere, right? Some areas still had that bone loss, right? Like very minimal, but it was only in certain areas. Like there's a term I use called furcation, which is right in between the roots, right? Those areas had a little bit more bone deposits or hard tissue.
And do you have any thoughts on why that is?
What we know now is I think it's the underlying kind of immune biology that's happening around the gum tissue. So there's a really neat paper published by Rich Darvo, and I think Camille as well, but where they showed that depending on where you're at around the tooth, like neutrophils, the density and the signaling of that neutrophil is very different, right? Even along that same tooth, right?
Interesting.
So I think it has to do with the underlying kind of immune biology that's happening around those areas. Also the microbiome in those areas, but obviously in mice, we're not going to be able to detect it. But so what we were able to see is that the same animal treated with rapamycin had this kind of gain of bone in certain areas, right? And to kind of add on to that, and that was the longitudinal section of it, the mice in Seattle replicated exactly kind of what we found in our first publication, but the attenuation was a little bit bigger, right?
Probably because the dose was
bigger. Right. Yeah. So in both sites, both studies, same dose, we're seeing kind of the same trend where the attenuation could potentially just be regeneration.
But it's more than attenuation. I mean, I think that's the thing that the Jackson cohort told us, right? Is there actually is regrowth of bone, at least in some areas, just from this 8-week treatment with rapamycin. So I think that's to me, that's probably the biggest discovery that came out of this paper. Okay, so what else did we look at in this study?
So I basically approached it like any dentist would, right? So I see bone loss, right? So then periodontal disease is also characterized by gum inflammation. So you could think about, and so there's gingival inflammation or bone inflammation that happens, and there's also microbiome changes that are tied in with that.
Right, and so gingival inflammation is when it reaches a clinical threshold, gingivitis?
So it might get a little bit cattywampus here a little bit, but so if you have clinical inflammation, so that's visual, right? Sometimes the gingiva is inflamed, but it's at a molecular level inflamed. So you might not actually see it. So when we talk about periodontal disease, we think about those images online where there's like bleeding gums and all this plaque. Many times it's not like that. Many times it's the gingiva looks okay on the outside, but when you take an x-ray, there's a bunch of bone loss. And when you actually look at, for example, the fluid around those pockets, there's high levels of inflammation.
So is gingivitis, though, that's when you can see the inflammation? Is that, I'm just wondering how it's actually diagnosed.
Clinically diagnosed, So if you have active inflammation with bleeding, so when you're probing around and you don't have bone loss, that's gingivitis.
Got it.
If you have a bone loss, then that's when it turns into periodontal disease.
Okay, thank you.
So for us, we didn't see any like big, like gingivitis, like red bleeding gums in animals. But so looking at inflammation and microbiome were the two other components that would really kind of tie this together in terms of periodontal disease with that. And so we looked at kind of both aspects.
And what did you see?
So with the inflammation, with the age-related mice, and we've seen this with other studies too, where that increased inflammation in the gum tissue with rapamycin gets decreased.
Right. And you looked at inflammation in the bone as well, if I remember correctly.
Yeah. In the same thing, specific inflammatory related markers in the bone also decreased over time. And what really kind of gave me, I mean, even with the bone loss data, I was convinced, but what really convinced me was the microbiome data, because the microbiome data, you know, we know that the rapamycin, I mean, this is in the food, like we talked about, it's encapsulated.
Right.
It's not, it doesn't get out in the mouth.
Right.
It gets distributed in the small intestines, I believe.
That's right. Yep.
And so it's systemic, right?
Right.
And so we knew that, and even in our data, we show that the old animals have a complete different microbiome.
Right. And I'm gonna double click on that just to reinforce what you said, because I actually think this is an important point that we maybe don't know the answer to yet. But in this experiment, because of the way the rapamycin is formulated, it's in this enteric coated microencapsulation. The expectation is that none of that drug, even though it's in the food, actually gets directly on the oral tissues.
Right.
It is designed to go through the stomach acid and then be released in the small intestine. So we don't think there's any direct effect of rapamycin on the oral microbiome. It's because rapamycin is presumably getting taken up and getting into the circulatory system. Now, it could get delivered back to the oral tissues, but we don't think it's a direct application to the mouth itself.
Right.
Okay.
Yeah. So it's a systemic dose that's not actually directly affecting the oral microbiome. And so,
and just for people who may be wondering if they know anything about rapamycin, we wouldn't expect rapamycin, even if we put it right on the oral microbiome, to have any impact on the bacteria there, because bacteria don't have mTOR, which is the target of rapamycin, but there's all sorts of fungal species in both the gut and the oral microbiome, when we know rapamycin is a potent antifungal. So it certainly could impact the microbial composition through its antifungal effects.
Right. If it's a directly applied on there. And so with the rapamycin treated animals, what we saw was that not only they had a complete different profile than the old, so the old were treated with just placebo, but the microbiome actually looked very similar to the younger animals
after
rapamycin treated, right? And so that right there, I mean, if our thought is correct, where there's no direct impact on the microbiome with rapamycin, showing the regeneration of that bone, showing the inflammation with a complete separate kind of organism that has a complete now different profile, I think that really kind of encapsulated kind of everything exactly what we were expecting it to happen.
What do you think is the mechanism for the apparent reversion of the microbiome? And I'll just say, it's interesting because in the study that Alessandro Bito did in my lab, where he looked at the gut microbiome, we didn't specifically look for reversion. I'm not sure that we would have detected it the way we did the analysis, but that didn't come out of that study. That it actually, we saw a difference between the young microbiome and the old microbiome, and then the rapamycin microbiome was kind of off somewhere else. It didn't seem to move that gut microbiome clearly, at least, back to a useful state, which was different than the case with the oral microbiome.
The one thing with Alessandro's paper, though, remember, I think we did it with fecal samples. So it wasn't directly. It wasn't directly inside the colon. So I mean, this is another thing that I know we talked about, which I don't think we got done. But looking directly into those intestines and seeing if those profiles are different.
And there you might actually expect to see more of an antifungal effect because of the release in the small intestine. So different design, different potential mechanisms there. Good point.
Right. Yeah. So and the microbiome, and to me, I keep going to the microbiome, is a lot of things we do in dentistry is because of these microbes, right? Dental cavities, cavity caries, right? Periodontals, there's some bacteria. The cleanings and things you get, is the idea behind it is the toxins that are released by those microbiome that causes these changes. If you can clean out those microbiome and have a clean kind of a platter, allows the disease to not progress, right? So,
And is it generally thought to be the case that the age-related changes in the oral microbiome are also more inflammatory? Because when we think with the gut microbiome, not necessarily that the microbiome itself is more inflammatory, but in part because of changes in the gut barrier, you get more microbial antigens that escape that and get into circulation, induce inflammation. But you could also imagine that if there are new microbes or microbes showing up with age in the mouth that are, at very low prevalence or in different places, that could actually induce an immune response.
No, and that's certainly I know there's groups that are working on that, like not specific with age, but there are groups working on that. I mean, the other aspect to this is not just the microbiome, but also the tissue, right? Everybody's going to have some sort of different microbial profile. But to your previous point, yes, it is going towards this kind of pathogenic potentially. Exactly what actually turns it into a clinical outcome, like I don't think we know yet.
Has anybody done microbiome transfers in the mouth where you take like a young microbiome and put it into an old mouth?
They must have. It sounds really simple to do, but I don't. Oh no, actually, no. They did. They have done that
in mice.
Well, it was actually in germ-free mice. So they were
okay.
So they transferred kind of the regular mice that you see in the facility. They're called specific pathogen-free mice or SPF mice, and they have a certain microbiome. And so one of the studies they show was actually transferring some of the SPF oral microbiome into the germ-free mouse to see if it kind of.
And just for our viewers, germ-free, what do you mean?
Germ-free means that they're well, one is it's really expensive to do, right? But they're encased, and basically there's absolutely nothing that goes in there. So they have no gut microbiome.
They've been cleared out.
They've been cleared out, and the facility is airtight.
Right.
And that's why it's really expensive to do. And so in that study, they were able to kind of transfer the oral microbiome into those germ-free animals to look at kind of periodontal disease progression, and really kind of see kind of what kind of oral microbiome microbes actually cause sort of disease. And so I know they did a study like that. In terms of young and old,
I mean, it's interesting, right? So there's, again, quality of the data, I'm not completely convinced, but there's some reason to believe that you can do this sort of microbiome transfer in the gut, these fecal microbiome transfers from young to old, and have some benefits. It would be interesting to know whether the same thing's true in the mouth. Okay, so we kind of talked about the oral microbiome. I think one question we weren't really able to answer here, and I don't know if you've got a feel for this yet, is whether or not mTOR inhibition. So again, rapamycin is an inhibitor of mTOR. So I think we're very confident the mechanism is going through mTOR here, but whether mTOR inhibition systemically is driving these effects or locally in the mouth, right? And I know lots of people have asked you, lots of people have asked me, what about rapamycin toothpaste?
Right.
And I think the answer to that question, we need to know that answer before we have any idea whether rapamycin toothpaste is going to work. So is it because of rapamycin getting to the mouth, presumably in our case through the circulatory system, or is it because of rapamycin acting somewhere else in the body that's driving this? I mean, what are your current thoughts?
I think it's a systemic response that's happening, because if you think about it in terms of aging biology, this makes sense. I mean, we know, and I don't know if this is going to go into that route, but we know plenty of off-label use of rapamycin.
Sure.
Right. And we know people have, these positive experiences that they're encountering. I mean, obviously we got to do a clinical trial to actually validate some of those claims, but especially with their mice studies, and it really ties into the fact that by inhibiting mTOR, whatever that's doing systemically, and is showing up in the mouth, I think that's kind of the mechanism that's actually going to happen. Yeah.
And do you think that's immune mediated, or?
I think it's most likely immune related, because one of the major components of periodontal disease is those kind of immune dysregulation, and that's one aspect and that's age related, right? And that's an aspect that never gets targeted in the current therapy. The only thing we target in periodontal disease right now is that microbiome, right? And just from our study, it's not just microbiome. There's inflammation that happens. There's bone loss that happens. There's all this other aging biology that's happening that we probably haven't even uncovered.
And so you think that is driven, at least the inflammation and perhaps the bone loss is driven at least in part by infiltration of immune cells into those oral tissues?
I think it's a lack of. So it's not able to cope with the ability to kind of fight back
against the oral microbiome.
Against the oral microbiome, or even and because of decline in the immune dysregulation of the gingival tissue in the bone. Right.
And at the same time, you're getting higher sterile inflammation, right? So it's this dysregulation,
right.
Which again, we know in other tissues, rapamycin is very potent at sort of restoring that balance.
It's almost like reset. I don't know if I would say resetting things.
I mean, I think that's a reasonable way to think about it.
So it's resetting kind of the inflammatory mediation, so that now you're able to cope with whatever's happening better.
So, okay, so at the end of this study, right, I think we knew, or at least we were very confident based on the data, that rapamycin treatment could partially reverse age-related bone loss, very potently, apparently reverse age-related increase in inflammation, and at least partially revert the oral microbiome back to something more like a youthful microbiome. Right. Okay. So is there anything you've learned since then that you think is important and cool here?
So one thing we're finding out is that those kind of positive effects that's happening with the periodontal is being persistent. So there was another study that we did right after this was so the idea is, okay, so after 8 weeks, we have all these positive changes. Right? So even with Ellen's study we talked about earlier, there's cardiac function improvement, and we see periodontal. So after 8 weeks, what actually happens?
Yeah.
So if the medication gets stopped, right, after a while.
Yeah, how long does it last?
How long does it last, right? And so we designed a study where, we gave the animals 8 weeks rapamycin, and then we took them off and then let them go on kind of the placebo diet. And what we're finding is that the inflammation markers, it's actually persistent. So it's actually even persistent even after 8 weeks. Now you could argue like maybe we have to take it out longer, but at least we know that the short term is enough to carry out the decrease in inflammation even 8 weeks after.
So at about the same length as the treatment,
the same length of treatment.
And did you go long enough to see when they come back, or do you have a feel for that yet?
So we did write a grant on that. So again, I mean, maybe it's just dentistry and aging biology hasn't quite merged yet, but we're going to probably resubmit
seeking
that.
funding.
Yeah, seeking funding. Yeah, but that would be another question, right? So at what point does it come back up?
Yeah.
And that could definitely alter kind of some of the clinical trials that are happening, right?
And also what happens if you do like cycles, right? Maybe it comes back in 16 weeks, you do another 8 weeks. Yeah, all sorts of interesting stuff there. What about the salivary gland?
Yeah, so another...
this is something that I'm really interested in, and we never actually nailed that.
We never published it, but what we found... So not only periodontal disease, but we also looked at kind of salivary function, and you could do that in mice.
Right.
We know that in old mice, the salivary flow decreases with age.
And that's true in people as well, right?
Right, exactly. And if you look at histology of salivary glands, there's like senescence, there's atrophy. So we could actually quantify the salivary flow. In a subset of our animals that were treated with rapamycin, we actually looked at, okay, how does the salivary flow change? And the salivary flow actually increased. And I know you've seen this data where in the animals that were treated with rapamycin, there was actually an increase in salivary flow. And really the question was, okay, why is this happening? Like, what are, and it goes back to kind of the science, right? Like, what is the mechanism of why this is happening? But it's actually increasing salivary flow, at least in mice.
Yep. And also reducing inflammatory markers.
Exactly, right, in the salivary glands. Right?
Yeah, and I don't want to get too far. I assume at some point you would like to publish this data, so maybe you don't want to give too much away, but if you're comfortable talking about it, like, what do you think that inflammatory signal is coming from in the salivary gland?
It's a combination of various tissues in there. So, salivary glands are always stored in there. It always gets circulated. And so there's a variety of tissues in there. And I think some of the inflammation is coming from actually senescent cells from the salivary glands. And we're actually seeing that, histologically and time-wise. And that is probably driving some of the inflammation that.
Right. So again, aging, we know one of the hallmarks of aging is increase in senescent cells. We know senescent cells give off a strong inflammatory signal, this senescence-associated secretory phenotype. And so you see that in the salivary gland, and we know even from other studies, rapamycin, again, it's very potent at knocking down that inflammatory signaling in senescent cells. It's called a xenomorphic. It doesn't kill the cells, but it knocks down the inflammatory signals. That makes perfect sense. And again, this just makes, I cannot believe that you've submitted grants on this and they haven't been funded. And again, I'm not dependent on NIH funding, so you don't have to say anything, but it is just ridiculous that we don't know the answer to this because some reviewers don't seem to think it's important. It just makes no sense to me. So if there's anybody listening who would really like to fund cool, important biology, it would be great to know, how is rapamycin impacting salivary function, and does it do that in people? Because if it does, that's a big deal. So anyways, it's very frustrating to me that you haven't been successful at getting these grants funded. I'm not blaming you because I've had lots of grants not funded.
Sorry, I don't want to keep going on, but like I still remember one of the comments I got back, or maybe you remember, is because mice don't get ulcers, it's not a good model for rapamycin. I think that was one of the comments we got. Like, because they don't get ulcers.
Because they don't get the mouth sores.
They don't get the
and
mouth
actually,
sores.
we don't know that's not that's
I don't
true
even know either, right? But we're
yes.
like, okay, well, so
there's quality review at NIH.
Yeah,
Thank you very much. Okay, so let's talk about humans, right?
Okay.
So because obviously that's of interest to many humans. What do we know about rapamycin and its effects on oral health in humans? And I mean, let's start with the sort of, anecdotal stuff from people who've been using rapamycin off-label. You were a co-investigator on the study where we published the paper last year, collecting data from, I think it was 333 people using rapamycin off-label, and then about 150 people who'd never used rapamycin. Right. We didn't look specifically at oral health there, other than the only side effect that was higher in the rapamycin users was mouth sores. I think about 15% of people experienced that. But you've been since looking at some of that data and exploring that. So to the extent you're able to share, like what have you learned from that?
Yeah, I mean, maybe I think this is a good time to kind of talk about mouth sores a little bit. I don't want to go too much into it because, when I first joined your lab, or when I'm in this field, mouth sores always comes up
with rapamycin.
With rapamycin,
because that's the most common side effect seen in organ transplant patients.
So to be clear, patients who are taking mTOR inhibitors, rapamycin or everolimus, et cetera, who had an organ transplant are taking very high dosages very frequently to suppress the immune system. Right. And as a part of that, one of the side effects is mouth sores. But
you must see mouth sores all the time as a dentist, and lots of people who aren't taking
rapamycin. Right.
And
And I'm not saying mouth sores are painful, right? So those patients who are getting those kidney transplants are getting the mouth sore. There's a specific term for that. It's called MIS, mTOR inhibitor associated stomatitis. Right.
Okay. And stomatitis is just another word for
mouth sores,
but of a higher severity.
It's like a higher inflammation. And when you look at photos of MIS, you're like, this is really bad. And that causes oncologists and physicians to decrease the dose in kidney transplant patients.
Right. And I think I've heard it's often the dose limiting toxicity, meaning people, they're so bad that people don't want to take the drug anymore.
And we're actually doing a separate study with the oral medicine department to see, and this is a complete side thing, but, those patients taking, these high dose mTOR inhibitors, where's the trajectory going? Like, what is actually happening? You dose reduce, does it actually go away? Right. So there's some other studies that we're doing. But so back to the point of mouth sores is that we knew from our study in the off-label use that people have been taking this continually for 2 years. Some people, right? If people were taking this off-label and they were getting MIS, like they should not be taking, they would not have continually taken it. Right. My theory, and because I don't personally take rapamycin, but from what I've seen or what I've kind of visually seen, is they're more like canker sores. They're more like little small mouth sores that will go away, and that's easily manageable.
And you're saying that that's the case in people using it off-label, presumably because either the dose is different, or the schedule is different, or organ transplant patients are taking a bunch of other drugs.
Exactly, right. Okay. So, and also higher dosages, right? Very high dose. And so one thing we're following up with the study that we could share is that most of the ulcers that people are reporting are not dose dependent. So just because you get a higher dose doesn't mean you get more mouth sores.
Interesting.
And the reports that we get that people are reporting is like they're calling it persistent ulcers. So maybe at the beginning they're getting some, but it just goes away on its own. Right. And so it's not that, the
extreme,
stomatitis, mucositis, right?
Yeah.
And so I think one plug here is if you're going to take it off label, like talk to your dentist, because they have ways to kind of manage that. If you do have those really bad sores, there's ways that we can manage it really easily.
Yeah. Now let me just make one point about the way that we collected the data in that study is that it is possible. So again, most of the doses are like 6 milligrams once a week, plus or minus 5, right? I mean, people are kind of all over the place. And I think what you said is you don't see any evidence from that data that the dose itself is driving the likelihood of mouth sores. And the severity doesn't seem to be at all severe in that group. And I think the point that I want to make is simply that it's possible that there were people who started taking 6 mgs of rapamycin once a week, had really, really bad mouth sores. They stopped taking it and they didn't take our survey because they didn't think that they could or should, even though we made the survey open to everybody. So it is possible that we didn't really have the right group to answer that specific question. This is always the limitation to those kinds of studies. What you really need is a clinical trial. So let's talk about the clinical trial. So you got a grant from the Impetus Grants program to start a clinical trial, double-blind, placebo-controlled, randomized clinical trial of rapamycin for periodontal disease. So take us through kind of what the goal is here and kind of where you're at now.
So the goal is to really just test, does rapamycin help patients with periodontal disease, just kind of mimicking all the mice studies that you've seen. And really the design of the trial that we could talk about is, does rapamycin allow patients having periodontal disease and getting the treatment have a better treatment outcome? Right. So currently, when you get diagnosed with periodontal disease, you get deep cleanings and you get these dental cleanings to maintain. But if we give rapamycin, could that reset some of the immune kind of functionality so that when these patients actually get the cleaning, do they have a better treatment outcome? Or just rapamycin alone, is that going to be enough to kind of help alleviate some of the symptoms that they're having? Because rapamycin is not indicated for periodontal disease, we had a long haul process to get FDA approval.
And when you mean indicated, you mean it hasn't, it has not been approved by FDA for that condition.
For that specific condition. So for us to conduct a trial at the university, et cetera, the FDA pretty much has to approve what's called an investigative new drug application, so an IND. So basically, they need all the data to show, we need to show them that this is safe, that we can get this done.
Even though it's approved by FDA, and in principle, a drug is safe, can be used safely if it's approved by the FDA.
And this is where, and you and I had discussions about this for a very long time, is I think this is where kind of that translational jump that our field, the aging field, has to make, has been challenging in a certain way, but also why we need it more. Is that we know the science is there. Like, you talk to anybody, whether it's rapamycin or the metformin, the science is there, right? But to get it into humans in a controlled trial that the FDA has to that the FDA needs to get involved with, that all that science, kind of goes out the window because there's no clinical data that's been approved to show that it actually is it's actually safe in healthy people, right? So from the FDA's viewpoint, rapamycin is still this immunosuppressant, right? Even if we published the off-label use, right? So it took a long time.
So what you're saying, though, I think, is that FDA is unwilling to allow any risk, or at least has to be a very, very low risk in people who don't have life-threatening disease. And periodontal disease is a disease, but they don't feel like the potential benefit of reversing periodontal disease outweighs the very, very small risk of using rapamycin.
Exactly, And
because they're uninformed, in my opinion.
And so that's why it took us over a year, multiple meetings, multiple renditions of our protocol, working with the FDA, trying to figure out a good medium to get this done, because at an academic institute, we need that IND to actually get this trial going.
You've got to follow the process.
All that process. So so after almost a year and a half of kind of working with them, they finally gave us the IND approval to do this trial. So we kind of, I call it, we got kind of the half of the golden ticket, right? Where now, yes, we're looking at periodontal disease, but we can actually test a lot of the questions you and I were asking during this whole kind of episode on, what is actually rapamycin doing? And so, so this trial, yes, it's for periodontal disease, but we're actually going to be looking at a lot of other kind of questions that our field is interested in, has been interested in, and really testing some of these hypotheses out at the same time.
That's great. So, now, I don't want to spend a ton of time on this, but you're not able to go and do the clinical trial that you wanted to do, right? Which I think was 6 milligrams once a week. So you're doing and that's because FDA is requiring you to do what's called a dose escalation. So you have to test. How is that working?
So basically what we need to show is that the dosages that we want to get to, which ideally would think it's going to be 6 milligrams per week, or is that we need to show that the doses before that is not going to make periodontal disease worse, and it's not going to be unsafe to the participants
Right.
in the trial.
Okay.
Even if you and I know from our label survey and many other kind of off-label use that, it should be fine. But FDA needs that data because per them, they don't have that.
Right. So you're starting at what dose?
0.5.
And then you're going to go 1,
1,
2,
2, 4.
Okay.
And what is really the reason why we got excited about that, because obviously this is not what we wanted to start off with, but once we kind of let the public know that we're going to do this trial, we already have over 100 people in the website wanting to be part of this trial that have never taken rapamycin or interested in taking rapamycin, who have periodontal disease. I mean, some people are writing me stories like, I've had periodontal disease for years.
I get those emails too. Yeah, absolutely.
And so to me, I mean, and this goes back to kind of the basic, right? We have to do the science correctly from the beginning. So part of us is like, yes, we have this little hurdle we have to go through, and we're going to go through it pretty quickly. But this is going to be data that I feel that the field can actually utilize.
Absolutely. And I think there is value in jumping through these hoops, right? And like you said, doing it the right way. So so that's fantastic. So you're recruiting and enrolling into the dose escalation right now, and then once you get up to presumably 6 milligrams a week, you'll do the trial.
So what we're going to do is right now, we're actually going to go higher dosages just from our off-label use data. So we're actually going to keep escalating up, right? But once we hit that 6 milligram per week, we're going to jump and start that placebo-controlled trial. And we have an NIDCR grant going in because of that.
So I was going to ask, because the impetus grant, while it was fantastic, and I as you know, I'm a huge fan of the impetus grants program. It's not a lot of money, So you're working on getting additional funding for this trial, to do the full trial, and so you've got an NIDCR grant in, and then is there a place where potential donors could come and donate to this trial? Because I mean, part of why I'm taking people through this process is, oftentimes I get asked, well, why hasn't anybody done this? And in part, it's because it's expensive, and in part, it's because it takes a really long time, and it is a lot of work to write all these protocols up and get the approvals. So if people are interested in donating to this research, is there a place where they can go and donate?
They could go through our website or go through the lab website that I have linked. I mean, to be straightforward, I mean, one thing that the longevity impetus grant allowed us to do was do this, like in their whole goal is to do this very efficiently. Right. I mean, you and I both know the grant system, right? Like, we submit a grant now, it doesn't get funded till a year later,
18 months,
and then we don't even know they're going to say no. So, hypothetically, if we can get the donation, we may not have to go through NIDCR, right? But NIDCR works right now because we've already done all the legwork that are major hurdles.
And it should get funded. I mean, don't get me wrong. You should apply for it. It should get funded, but we all know how that process works.
So for us, hypothetically, if we don't have to go through the NIDCR, that would be even perfect, right? Because then we don't have to kind of wait. I mean, if we submit the grant like in February, for example, we know the whole, and maybe people can look online too, but it doesn't get reviewed till like July, and then the funding decision comes out in September, so you actually don't start, right? And at that point, the momentum is lost. So, so
we'll post the link to the website where they can donate directly to this research. Great.
And we have everything set up. We're pretty much giving NIDCR a platter. Like, we have the FDA approval, we already have the placebos made, right? We already know what dose we're going to do. We have the protocol already set. This is a great way to give a non-surgical method, a very novel method to help people with periodontal disease, right? So we're serving it on a platter, right? So anyway, so we're doing the escalation, but we're going to continue the escalation beyond 6, right? We're going to still get FDA approval to do that.
Awesome.
And then with the 6, we're going to jump off and start the placebo, and then we're going to keep kind of doing this, and then maybe do another placebo, and kind of kicking it off from there.
And I think that that's an important question too, because we really don't know much about the dose response, and 6 is sort of an arbitrary number that, for reasons we won't get into, is where most people landed.
And we'll be asking, one of the primary questions, like, okay, for example, mouth. Is that really, how is that going to be maintained? But there's a lot of things in there that we packed in with a lot of other kind of scientists, kind of in the back end that we want to evaluate and to kind of see.
Great. Yeah. Well, I'm super excited to see what comes out of that trial. Yeah. Okay. So is there anything we haven't talked about? We've covered a lot of ground. Anything we haven't talked about that you feel, you really want to get off your chest here?
Yeah. Well, I think with this YouTube channel and kind of just in general, where the public is being aware of kind of longevity science, aging science. I mean, I think for somebody who is brand new, right? I've only been here for 8 years, maybe 9 years or so. And so I'm relatively new, right? But I have patients coming into the dental clinic asking me about these longevity treatments,
right?
About their impact on oral health. So people are thinking about it, right? One thing, and we keep going back to it, but people have to understand that there is strong science and there is not so strong science, and you call it there's snake oil things, right? And we're at this cusp where everything's going translationally, right? Even our trial, there's other trials that's happening, where we have a really good opportunity to show the community, whether it's research or the general public, that by targeting the biology of aging, that's the way to do it, right? And
21st century medicine.
Yeah, that's kind of what we're stuck in, right? So, but this is a critical time where we just have to be very, very cautious on, kind of the science that we report, on the public that we kind of send out. I mean, you and I both know we talk about this too, but we have an obligation as scientists to make sure we present the strong science and have a discussion. And it goes back to kind of how I was trained, right? If the science is not good, right, you call it out and we talk about it and we try to figure out, because at the end of the day, right, the goal for aging biology is to help people, right, optimize their health, right, optimize their oral health, right? I mean, and I think that's the ultimate goal. And because I am surprised at, patients in the dental chair coming to me with these longevity and asking me,
what dose, hearing about it.
Yeah, what dose do I use? What do I? I'm like, you don't even know what dose you need to use, and you're going to take these medications, like just because somebody said this, or take this supplement to say that. Like, as a dentist, if I'm hearing that, like,
yeah, and I mean, it's
it concerns me.
As you know, I agree with everything you said, but I mean, it's a challenge, right?
Yeah.
Because there are people out there who are less rigorous in their approach to science. And there's also a big gray area. I mean, I think we do have to be honest.
Right.
In this field, there's a big gray area where there's some evidence to support, but, we don't know for sure.
Right.
And even with rapamycin, I think you can make the case, we don't know for sure, but there's some pretty good evidence to suggest that it may actually have some benefits. So
there's a lot of evidence.
Yeah, there is. But you have, I mean, one of my point is that I think, there is this big gray area, and it's in that gray area that I think honest people can disagree, but I think we should also be honest that it is a gray area, right? There are probabilities and very little certainty with some of these things.
And especially now with things, because I don't know how many clinical trials were happening when you were going through, your postdoc or whatnot, right?
Not in aging at all.
Right, exactly. Yeah. And now we're at this point where that's that kind of now trend, right?
Yeah.
So this is where I think, the strong science is going to hold up, right? And not so strong science is maybe not going to hold up. And sure, there is that gray area, but I think, people listening to this channel or other channels as well, I think you want to, you have data out there, right? Read for yourself, understand what's actually happening, and
watch the OptiSpan YouTube channel.
Yeah, and watch the OptiSpan YouTube channel, because we'll have some other content.
We're doing our best to keep it real when it comes to the science.
Yeah. So and take care of oral health, right? Ask dentist questions. We now have technology to look at a lot of things. So, optimize your oral health.
Great.
Right.
Great. So that's a perfect place to end. So thank you so much, John. This has been fun, and it's been a great discussion. And thank you for watching the OptiSpan YouTube channel. As always, please leave comments or questions. And if there's any topics you would like to see us cover, if you'd like to see John come back, leave that in the comments as well. And we'll do our best to make it happen. So that's it for now. See you later.